This is a review of Chapter 4, Cell Membranes and Transport. The fluid mosaic membranes. Number 1. Describe the fluid mosaic model of membrane structure with reference to the hydrophobic and hydrophilic interactions that account for the formation of the phospholipid bilayer and the arrangement of proteins. So first thing is hydrophobic versus hydrophilic and what all of that means. So hydrophobic is water-hating, non-polar, but in this case it's also lipid soluble. I mean, that's always the case, but that's especially important when you're talking about the fluid mosaic model, the cell surface membrane. And then hydrophilic, water-loving, polar, non-lipid soluble. So, the phrase fluid mosaic model. The word fluid is in reference to the fact that both the phospholipids and the proteins can move about laterally by diffusion. I'm going to say it again. Both the phospholipids and the proteins can move about laterally by diffusion. That's important. And then the word mosaic describes the pattern produced by the scattered protein molecules when the surface of the membrane is viewed from above. The bilayer is made up of phospholipids. Phospholipids have a hydrophilic, polar, water-loving head, and a hydrophobic, non-polar, water-hating tail. When placed in water, the heads will face outwards towards the water and the tails will face inwards, creating a liquid-free environment like inside the cell. Not inside the cell, but inside the membrane. This membrane acts as a barrier to most water-soluble substances because water-soluble substances are not lipid-soluble, and it would have to be going through the lipids. And if it's not lipid soluble, then it can't get through. Proteins may be found in the inner layer, intrinsic, outer layer, extrinsic, or throughout transmembrane, the membrane. Proteins have hydrophobic and hydrophilic regions. These proteins stay in the membrane because the hydrophobic regions, made from hydrophobic amino acids, are next to the hydrophobic fatty acid tails and are repelled by the water environment on either side of the cell. The hydrophilic regions, made from hydrophilic amino acids, are repelled by the hydrophobic interior of the cell, thus face toward the aqueous environment, either inside or outside of the cell, or they can make up channel proteins, as in they're facing the water-filled pore. So with proteins, it's basically just they have hydrophilic portions and they have hydrophobic portions, and those portions are going to stick with the part of the membrane that corresponds with that. So the hydrophobic part of the protein is going to be with the hydrophobic part of the membrane. Hydrophilic part of the protein is going to be with the hydrophilic part of the membrane. 2. Describe the arrangement of cholesterol, glycolipids, and glycoproteins in cell membranes. Cholesterol has a hydrophilic tail and a hydrophilic head and a hydrophobic tail, and spans the membrane. So it's just in between the phospholipids. Glycolipids and glycoproteins form the carbohydrate chains that branch out from the proteins and lipids, the glycocalyx. So on top of the membrane, you got your little lipids and your proteins, and sticking out of that your carbohydrate chain, and that is made up of glycoproteins and glycolipids. Number 3, describe the role of phospholipids, cholesterol, glycolipids, proteins, and glycoproteins in cell surface membranes with reference to stability, fluidity, permeability, transport, carrier proteins and channel proteins, cell signaling, cell surface receptors, and cell recognition. So phospholipids, the fatty acid tails may be saturated or unsaturated. remember that saturated means like fully saturated with hydrogen bonds all the way around, and then with not hydrogen bonds, sorry, double bonds, and then unsaturated has like the carbons. Unsaturated tails are bent, meaning they fit together more loosely, which in turn means the more unsaturated the tails are, the more fluid the membrane, because they're all bent up and they're all and then another important thing with phospholipids is that as temperature decreases, the membrane becomes less fluid, which is something that cholesterol can fix. Cholesterol. Animal cells contain almost as much cholesterol as phospholipids. Almost as much. Cholesterol is important for the mechanical stability of membranes as it reduces fluidity. So it stabilizes it and makes it less fluid. The hydrophobic regions of cholesterol molecules help to prevent ions or polar molecules from passing through the membrane. Cholesterol helps the cell surface membrane to maintain fluidity at low temperatures because of the stabilization. So in the same way that at regular temperatures or high temperatures, it's maintaining the stabilization by just standing up, by just being there, it's going to do the same when at low temperatures. So at low temperatures, when the membrane is trying to like squish together, I guess, in a way, the cholesterol is going to be there to stop it from fully collapsing in on itself. Glycolipids and glycoproteins, they act as receptor molecules which are important in cell signaling. They may also act as cell markers or antigens. I'm sorry. Proteins. Proteins may be present as transport proteins. Transport proteins show specificity to their molecular ion and may be present as carrier proteins or channel proteins. 4. Outline the main stages in the process of cell signaling leading to specific responses. Secretion of specific chemicals, ligands, from cells. Transport of ligands to target cells. Binding of ligands to cell surface receptors on target cells. So, with cell signaling, step 1, a stimulus causes a cell to release a ligand. Step 2, the ligand is transported to the target cells, typically through the bloodstream. If it's a mammal. If it's talking about a plant, it's not going to be the bloodstream. Don't say that. Just say transport systems. That's a broad enough term, but still specific enough that you're allowed to use it. And then step 3, the ligand binds to the receptor that it is specific to. So again, we're talking specificity. The receptor, the spot on the receptor is specific to the ligand, and the ligand is specific to the receptor. Step 4, the receptor conforms its shape, allowing it to send a message to the G protein. So the process in which it's changing shape in order to release a message, that is transduction. And then step 5, the G protein acts as a switch to bring about the release of a second messenger. More specifically, so remember that phrase. The G protein acts as a switch to bring about the release of a second messenger. I hear talk that Cambridge loves that phrase. But more specifically, the G protein sends a message to the enzyme, which sends a message to the second messenger. And when the G protein is triggered, that is the beginning of the signaling cascade. Because so much more happens after the second messenger receives the message. And that's all for 4.1. I will go write 4.2 and then be back.